I really appreciate the opportunity to be able to speak with you and ask you questions because I have so many questions I've been wanting to ask that are really science related. And the company's done so much from a commercial point of view and financially fixing its balance sheet and that kind of thing. But I would like to get into the science because I think your platform is phenomenal. And this gives us an opportunity to sort of demystify what is going on in synthetic biology and what you're doing. So with that as a background, I was going to ask if you could maybe give us a simple definition of what is synthetic biology and where is the industry today?
Mm-hmm. Yeah, so synthetic biology is what we really term the field of biotechnology today. I mean, this is, it's not a new platform. Humans have been using fermentation-based biotechnology for a while now. I mean, one of the most famous biotechnology products was, of course, insulin, which was made through fermentation.
Right.
But today, the tools have evolved drastically. And so what we used to call the field of biotechnology can now be referred to as synthetic biology, in the sense that we can use a lot more synthetic DNA to rapidly engineer our strains. But from the standpoint of what the end result is, it's the same as what the industry has been doing in biotech for a while now.
Right.
The only thing I'd add, Sunil, just a couple of references that I think could be useful for folks. So first, there's a long history in biotech, right? Biotech, as we know it today, really got developed or scaled quickly by Biogen and Genentech. So think about that as what we know as biotech today really emerged in a rapid way in the era of Biogen and Genentech. Genentech at one point had a portfolio that was very similar to ours, everything from industrial to pharmaceutical, and then decided to really focus in pharmaceutical back in the mid 80s. The other way to think about it is kind of related to the semiconductor industry, where everything from the microchip to the operating system created the infrastructure to really enable the semiconductor computers and then the internet to really thrive. And if you think about where we are today, you could think of it as there are the Intels of the world that actually provide kind of the equivalent of a microchip, and that would be something or somebody like the company We Buy DNA.
There are quite a few. I mean, Twist, IDT.
Twist Biosciences is a good example of the Intel of the industry. And think about us as the operating system of synthetic biology. We're the operating system, meaning we're the coders and have the code that writes and makes clean chemistry. And think about that as the role of synthetic biology is the ability through fermentation and biotechnology to make chemistry. So anything in the world that uses chemistry is the potential for synthetic biology to play a role in. And then the question is kind of like, what role does it play? And all that depends on the end application. So think about operating system, chips, and then the application. The application of the products we make, the operating system is really what we are, we code. And then the chips and stuff that we use, like the Intel inside, are like what a Twist Biosciences does, as a way to think about the landscape.
Right. Well, I wanted to try to understand from the point of view of the size, the potential for the industry to grow. Amyris is at the point where you're focusing on yeast fermentation. And I'm just wondering, of the estimated 326,000 molecules out there in nature that nature's created, how many could be made by yeast fermentation? And what is that process? What does it mean to be using strain engineering to design or redesign yeast strains so that you can produce a particular molecule? That's what I was hoping to get into understanding, so the investors and then the public can understand the industry. Thanks.
Yeah, I mean, theoretically, you can engineer yeast to make every single one of those 326,000 molecules you refer to.
Wow.
The question is yeast always going to be the right host to make those molecules? So
Yeah.
you asked a question, what is that process? What's the fermentation process look like? So yeast has evolved over billions of years to do one thing, right, which is consume sugar, convert it into ethanol. I mean, that's what beer fermentations and wine fermentations rely on.
Right.
Now, the question is, if I want to, instead of making ethanol, let's say I want to make molecule X, how do I then convert sugar to molecule X when the yeast is trying to make ethanol? And so now this is where we get into what are all the reactions that occur in any living cell. So living cells, all our cells, we function because our cells keep carrying out reactions at any given time, chemical reactions. It's like I like to say that nature is the world's best organic chemist.
Yeah.
Okay. Now, each of those reactions is, of course, dependent on a certain genetic sequence. And so if you'd like to now alter the reactions happening in yeast to make molecule X instead of ethanol, you need to change the genetic code within that yeast.
Right.
And so at the end of the day, when you alter that genetic code inside yeast, it consumes the sugar and starts making molecule X, which is your molecule of interest. That, in a very simplified way, is what the fermentation process is about. Now, of course, you need to fine-tune a lot of other processes because you're trying to fight billions of years of evolution, and yeast will always, or any microbe will always try to fight against what you're trying to do to it.
Right. So just to understand what nature's done and what, although the decades or really centuries, people have made beer and wine and all kinds of things with yeast, they had to find the particular yeast that would make the right kind of wine or the right kind of beer. They just had to do a hit or miss or try to find them in nature. And so what I'm trying to understand is that synthetic biology is actually helping to find a better strain of yeast and actually making that by using DNA analysis and reconstruction?
Yeah, so we would probably use the same chassis as, say, a beer making yeast.
Okay.
So we won't try to change that, but what we will do is try and go into that strain and alter some of the machinery inside that yeast. So it's the equivalent of, you take the same chassis of a car, but you put in a better engine or you modify certain components of the engine to get different performance or maybe even better performance.
So almost another way to think of it is we spent thousands of years roaming through nature to find the right organism. And what technology has done over the last 30 or so years has really enabled us not to have to worry about finding the right organism, but instead inserting genes, enzymes, and changing the genetic code of an organism so it does what we want versus us having to find one that does what we want. So that's really the biggest transition. And what synthetic biology has brought to the world is the ability to make an organism do what we want it to do versus having to discover one that does what it wants to do in nature.
Okay, so you've done, how many different strains have you been able to develop over time? And how is it? It appears to me that the pace of innovation has been accelerating in the last few years. So how, were there some discoveries that breakthrough discoveries or enabling technologies that helped you get to this point of developing so many different strains?
Yeah, I'll just back up just a minute here. So anytime you would like to take a yeast strain or any microbe and convert it into something that makes a molecule of interest, you go through this, what we call design the strain, build the strain, test it in the lab, analyze the results. And then based on those results, go back to the design process. So it's what we call the design, build, test, analyze cycle. And it's a very iterative process. Now, again, even a decade ago, going through that one cycle of that iterative process took a lot of time and effort. Now, the issue is biology is unpredictable, right? I mean, humans are trying to, so some control over the strain, but biology is unpredictable. So if you have only one chance to go through that design, build, test, analyze cycle, your odds of getting it right are quite low. And so what Amyris did, we realized very early on that we need to change the paradigm of how we approach the problem. And what we said is we have a higher chance of success by going through that iterative process thousands of times in a given experiment as opposed to a single attempt. And so what that allows us to do is test a huge space of testing and then
Yes.
find the right strain that we need for our process. Now, of course, how do you do that? Well, the way you do that is you get away from laborious manual processes in the lab. You start relying on robotics, and automation to actually carry out some of those experiments. You start relying on software engineering, machine learning to help you understand the data that's being generated. And so you can start using better tools to help assess the next strain. And of course, we had to also optimize the way molecular biology was being done. So, John talked about inserting a gene into a yeast strain. That's a very manually labor-intensive process. And so at Amyris, we developed tools to make that process, inserting the gene into yeast, as predictable and as efficient as possible. So when you combine these three, so cellular programming, machine learning, and data analysis, as well as better tools for testing and building strains, you end up getting with the platform that we have at Amyris today. So sorry, go ahead.
Yeah, so this sounds like a very complicated process, and I'm just wondering how, I was just wondering why is it taking the industry so long to get here? If in fact fermentation has been around for centuries, if in fact we know that you can find a yeast strain that'll produce such and such a molecule, it sounds like it's a very difficult and complex process. And I'm wondering how fast that pace of innovation is. Is there a way for you to tell us, how quickly you can find a new yeast strain relative to, say, 10 or 15 years ago?
Sure. I mean, 10 or 15 years back, it would take us anywhere from 3 to 5 years. So let's assume we were starting to work on a molecule. 10 to 15 years back, it would take us 3 to 5 years to go from the first strain to, let's say, the pilot plant for that molecule.
Wow.
Today, it's less than a year, right? At Amyris, we take less than a year to go from designing the first strain to where we are on the pilot plant. Now, why has it taken so long, right? What are the innovations that have allowed us to get ahead? Let me give you an analogy. Even as far back in the 90s, you take a road trip and you have to refer to a paper map and you have to figure out where you want to go from point A to point B.
Right.
Now, over time, you have companies that have accumulated a database of all the roads and highways and created a software tool. Now, of course, there's a lot of intelligence behind those software tools, right? Because you can't just say point A to point B. The mapping algorithm has to figure out the best route possible without sending you over a river where there's no bridge. Right. So in the same way, what we have done at Amyris is we have started cataloging all the reactions and all the genetic sequences that occur in nature, and then use those software tools to tell us, hey, what is the best way to get from sugar to molecule X? And those design tools then allow us to search a pretty large amount of space very efficiently.
I think, Sunil, what you just said is real important in that there's a combination of things, back to Graham's question, of enabling technology that has actually evolved in the world, everything from information technology, data management, robotics, artificial intelligence, that have all been enabling technologies to help us. On the other end, there are things that only we have, which is the mapping around every iteration, every strain we've ever engineered. We've collected data in every reaction that we've engineered and what the impact of that reaction is. That unique data is owned by us, and that evolution of the map, in other words, understanding the topography of yeast, has enabled us to be much more effective in every new molecule we engineer. That data and the history and the number of reactions, which is all about number of yeast strains we've engineered over time, data we've collected, actually gives us a competitive advantage that's only gained by someone who's able to replicate the number of experiments and has the data that we have. And I think that is a huge differentiator that we don't talk about much because we take for granted the maps that we've built. But the maps that we've built are deeper and more thorough on the yeast topography than anyone in the world, just by the fact that we've invested more and have engineered more than anyone in the world. Everything else is enabling technology that everyone else has access to. There are a lot of very smart people and a lot of companies in the field of synthetic biology using those enabling tools. But the data on the topography and the learnings about what the reactions are is something that's unique to the engineering that we've done to date.
Yep. I mean,
How long would it take? How long would it take for another company, one of these newer companies, or even some of the other larger companies like Genentech or Amgen or Biogen? Could they just enter this industry and say, "Okay, hey, Amyris is onto something. Let's do this." How long would it take for them? Could they do it? Could they
I
enter
mean,
the industry?
sure, you can do it. The question is how much time do you want to spend doing it? It's going to take years to do it. And it's not as simple as just buying the instruments of a vendor. The key to building this pipeline is integrating it efficiently with the right goal in mind. John
Yes.
talked about the data and the topography, of the reactions, right? We didn't even talk about the fact that Amyris is one of the few companies that not only has lab data that we apply to our databases, but also information from our manufacturing systems. We are, again, one of the few companies that has manufactured 11 different molecules at scale. Each of those manufacturing campaigns results in a rich data set that we feed back into our information systems.
Right.
So today, when we start a new project, we're starting with the end in mind, right? We're thinking about what does manufacturing look like, and our data systems take that into account. And so that is also a huge differentiator.
So you have a totally integrated process from idea to through R&D to strain engineering, enzyme development, product development, and then to going to scale. And then it's a feedback loop so that it helps you design the next molecule?
Perfectly put. Yes, it is. And that's the key. You might have every companies might have disparate parts that operate quite well individually, but unless you integrate them into a fully functioning pipeline, you won't gain the efficiencies that Amyris has had.
So how many years?
Here's a good example. One of the biggest problems in scaling a highly engineered organism is the stability of the organism. How well does it operate in a tank when put under all the normal conditions of large-scale manufacturing, large-scale fermentation tanks? I would tell you it cost us in cash about $200 million to get through the learning for us to be able to have enough insight in what is it about engineering an organism and what is it that we actually screen for, what we look for, that gives us an indication that we have a stable organism at scale, which has really enabled us, that particular learning has enabled us to take at least 3 to 6 months out of the development process, because we can go directly from a small scale 2-liter tank fermentation to a 200,000 liter large scale fermentation. I believe we are the only company in the world that's been able to replicate that over and over again consistently. That took $200 million of learnings and a lot of wasted fermentations until we were able to learn how to actually get to a stable strain for large scale fermentation. The fact that we had to operate at large scale forced us to learn that. And the fact that we had the capital to invest in those learnings enabled us to get there. We don't believe anyone else has been able to do that.
Yeah. So Amyris is quite clearly a commercial leader in the industry with 10 molecules you're producing and selling for revenues and producing in large scale. And you're now talking about tons, not just kilograms. And I'm just wondering how many other companies are producing anything at scale, whether it's one molecule or 2 or 3. Are you so far ahead that you're not only the first to do 10, but others are only maybe producing 1? It's hard to get data on competitors. Thanks.
Yeah, and it's actually, Graham, it's actually 12 now as of the fourth quarter, right? So I think the rate at which we've been scaling and commercializing, it's really been unbelievable in the last couple of years. And Sunil, you should take on the competitive question.
Yeah, so I mean, company our size, we are the only ones who've taken so many molecules to scale. There's absolutely no doubt about that, right? When you think about companies that have successfully scaled up molecules in fermentation, these are companies that have been around for decades and are far bigger than we are, right? But in our space today, Amyris is the only one that has commercialized so many different molecules at scale. And that's through the investment that we made in the in the innovation platform.
Right. Well, you also mentioned that Amyris has actually developed its own tools, and though to the extent that's proprietary or patentable, are there any Is this kind of part of your the moat around your IP, intellectual property?
Yes, we are
that
actually.
you have your own tools that others can't really use?
Yeah, so we have a very well-rounded IP portfolio where we protect the tools and the lessons learned right through this process.
Okay.
Some of it is going to remain trade secrets, but a lot of it is patented. Yeah, and so these tools are proprietary. We again lessons learned from the last decade of commercializing so many molecules doesn't come easy, and we make sure we protect our portfolio through patents, and in some cases we also have trade secrets that we don't disclose to anyone else.
Right. Well, I just I was lucky enough to be introduced to a biologist at one of the major universities, and he has remarked at how amazing it is that Amyris has been able to, what he calls, master so many different biologic pathways to produce so many different kinds of molecules. It's not that you're producing 10 molecules or now 12 from one pathway. It's a variety. So I was wondering if you could maybe help us understand what are biologic pathways or synthetic pathways or synthesis pathways, excuse me, and how many have you mastered, and therefore how many different kind of molecules does that enable Amyris to make in the near term?
Yeah. So when Amyris first started, our focus was really on the pathway to produce terpenes. So terpenes are a natural class of molecules. There are over 50,000 of them. And so the pathway to convert sugar to terpenes, we mastered that very early on.
Okay.
Over the last decade or so, we've actually embarked on learning about other metabolic pathways in microbes. And today we have expertise in more than 20 metabolic pathways. And we've actually commercialized molecules from other pathways among the 12. Now, so more than 20 pathways is where we have started where we have lots of experience. You asked how many molecules do we make? Apart from the 12 molecules that we've already commercialized, the 17 molecules that are under current development, we've actually produced more than 200 molecules in the lab.
You've used yeast?
Yes.
Yeast engineering to develop strains to produce 200 different chemical molecules out there already.
Yeah. So it's not just yeast. We've also diversified our expertise into other organisms. So these are, these greater than 200 molecules are spread across yeast and several other organisms that we have started engineering here at Amyris. But the point is that we are trying to cover such a wide space that tomorrow, if we need to go after another molecule, we're probably only 1 or 2 steps away from engineering a current strain to get to the new molecule. So it's a theory that it's what we call the platform strain. So we have platform strains for pretty much any metabolic network that's needed to take a molecule from design to commercial scale.
So you're saying that you have enough basic primary science understood and the pathways mastered that you can go, when you say 1 to 2 steps away from just about any molecule in nature, or just you're talking about hundreds, or you're really talking about thousands?
I'm talking about hundreds of thousands of molecules.
Wow. And by 1 or 2 steps away, meaning what, a year or 2 away, or
less than that? I mean, again, our record today is we can go to the pilot plant in less than a year.
What's really interesting is because of exactly what Sunil is saying, and the fact that there's a very good understanding about the capability of our platform across major industrial players around the world, we're actually coming at it from 2 different perspectives. One is for a developing market around plant proteins, that we're able to develop target plant proteins for some large partners who would like to be in that business. I think on the other extreme, it's actually making molecules from cows or other current protein providers to the world that those industries would like to have more of without having to grow more cows, or making sure the cows are more productive, right? So we're dealing with everything from making a molecule called heparin, which is a blood thinner, all the way to a natural preservative that makes meat stay fresh longer, to a molecule, a protein that can be added to meat to make it feel more tender as you eat it, right? So when we think about the market, for alternative proteins, we're not like single-minded about, well, we've got to be the next plant protein alternative. We're thinking about how can biology make chemistry that's available today more available and more sustainable. And that actually opens up the spectrum for us across both making current meat production more sustainable, as well as new sources of plant-based protein. So I just wanted to build on that the approach we're taking is not a single point. It's actually, there are 4 or 5 molecules, all of which can add significant benefit for the protein industry that we're actually engaged with multiple partners on. That'll be the next big jump for our platform, right? The last big one was getting into cannabinoids. The next big one is making proteins. And the one after that will be making antibodies that make therapies more sustainable for the world, right? And so that's the way we think about building on the point Sunil made, which is the platform is now very fast to market. And we have a tool set and a library that enables us to get to target molecules faster than anyone in the world. And we're using that to go into markets where we can create a lot of value quickly.
Right. Well, it appeared to me, and John, I've mentioned this to you in the past, that it appeared to me that it looked like you were entering larger markets based on your most recent announcements on new products. And your comment to me was, yeah, and more profit at higher margins. Is this by luck or by chance, or is this because you're able to drive your costs down just even in the last 6 or 12 months? Or is this because you're getting better at selecting more profitable, larger markets? How is it that all of a sudden you're able to be entering cannabinoids, HMOs, adjuvants for vaccines, even monoclonal antibodies, which I was amazed to find out that 7 to 8 of the largest 10 drugs in the world are monoclonal antibodies, and that you're developing ways to produce those cheaper. How is it all of a sudden these larger markets are at your doorstep?
I mean, I wish I could tell you it was all of a sudden. I can tell you from
it seems like it to me.
From the point we pivoted, which was back in 2011, 2012, when we decided to get out of large commodity markets, it was always our intention to get to higher and higher value. So that was always strategically where we wanted to go. And then around 2015, 2016, we started to see opportunities into markets that had small volumes, but much higher price points as the way we think about value. So even though the total size of the market can be bigger, it's typically bigger from a dollar perspective, but not a volume perspective. So what we ended up doing is transitioning out of large volume, very capital intensive, like small margin per product, to large value, big dollars per unit, like north of $1,000 a kilo price point versus south of $10 a kilo. So that's, a significant multiple on the capital we have employed for how we generate revenue. And a big reason for us being able to go there wasn't because we hadn't thought about that, but our initial movement away from commodities was really developing exactly what Sunil described. It was developing the understanding around these different pathways, the capability to be one step away from these different molecules, to then apply that capability to be in fast to market. And I think we just started to hit that stride in the last couple of years. And I think you've seen us gradually build that value, right? From going to HMOs, going to cannabinoids, now proteins, and then going into the antibody production. All of that an evolution enabled by capability of being faster or one step away from these molecules.
Yeah, I mean, it takes a little time to set up the platform. You build the pieces, and eventually you reach a point of exponential growth, even in the technology platform, right, where you can just explode. And today, the goal is to be market agnostic, right? The platform needs to be able to go after a market when it's available, and we believe our platform is there today.
So your platform has not only gotten stronger by getting deeper in the scientific platform, by getting deeper in the science, the pathways, the enzymes, et cetera, but the DNA analysis, but also it's gotten broader so that you've developed just about a lot of pathways, enough pathways that you can target just about any molecule. And that has enabled you to then be have the luxury of targeting more attractive end markets with higher value products.
Perfectly said. Perfectly said. I'll say, and the third thing is we've also gotten a lot smarter.
Okay.
So earlier, I think you would
That
need
helps.
to apply brute force,
Yes.
to try and solve a problem. Today, we have the tools. If we do need to apply brute force, great. But why apply brute force when you can apply the right force?
Right.
And so I think that's what has changed. We've all, because of all the data we've been collecting with every experiment we run, we are able to make more informed decisions. So we've gotten a lot smarter also in how we approach science.
Yeah, I was wondering if you could maybe touch on some of the molecules that you're already producing and what you had to master, and therefore how that opens up other potential targets that are currently perhaps in your pipeline of what I think it's 17 molecules are currently in the R&D pipeline, probably closer to the development stage, I would assume. And so if you could sort of talk about the science behind the ones that you've come out with already commercially and the ones that are in the pipeline and what that portends for the future. Thank you.
Yeah, so I mean, I can give pretty, most of the molecules end up going through nearly the same kind of problems that need to be solved. So John talked about a very important one, right? Stability. And so what that means is you start the fermentation tank and after 3 or 4 days, it stops producing the molecule of interest when really you would like to continue to do it for days. And so, on the technology side, we had to solve the stability problems, make sure that the strain stayed robust over the entire period of fermentation. So that required us to build some proprietary genetic tools that are now incorporated into every strain that goes into manufacturing. The other point that is, that we focus on a lot is every molecule we produce, let's assume there are 10 reactions from sugar to the molecule. Let's just take an example. Typically, at least one or two of those reactions are quite inefficient because nature has intended them to be inefficient. Nature didn't need them to be as active as we would like it to be.
Right.
And so we end up having to do a lot of enzyme engineering. And what that means is that specific reaction, how do you make it as efficient and as specific as possible? Okay, so that's, again, we have to develop a lot of tools in being very good at enzyme engineering. I think the third part is around improving the purity of the final product. And so again, at Amyris, what we believe is that the consumer is going to accept an ingredient when it's sustainable in its production, cost effective, and pure. Right. Now, so we have to keep a close eye on how pure the ingredient is that we're producing. And so there are a lot of challenges that we need to solve around that front. So I just want to touch on 3 main things that we do look out for when we go after any single molecule.
So
John,
it's almost
look,
I would, I think that's well said. And I would add this basic overview that the backbone of our engineering, which is all about stability and large scale manufacturing, we really developed a lot of those breakthroughs in engineering the strain to make farnesene as efficient as it does. I mean, there is nobody in the world that can make farnesene even near our efficiency. And then there are many breakthroughs, if I think about the enzyme for sweetener, if I think about some of the breakthroughs on cannabinoids, like every one of them has brought kind of a new iteration for some function that the strain has to do that's enabled us to expand our application in a new class of molecules that before we thought would be very difficult. So that's kind of the way I would describe it, like the backbone of our strain engineering and major foundational breakthroughs are all about farnesene. And then for every one of the molecules, we've had a breakthrough that's been unique, that's enabled us to access a whole new class of molecule that before was pretty complicated.
Mhm.
Right.
Yeah.